So she's going to be helping us with the program, and you'll be seeing a lot of Sarah
Yay.
with Kim. So become very good friends with Sarah. And welcome to a very exciting program. So we're going to start off with our first presentation, Stephanie Lynch. And I also wanted to introduce the fabulous mentors, Jackie Duncan, and we're excited to have Jackie. And also a shout out to Ellen Weber as well, who you'll hear about in a second with Steve.
All right, so with that, I'll take it away. So my title sounds very esoteric, but I promise by the end you will understand what it means. So my project is going to be looking at cone imaging in patients with retinitis pigmentosa who are going to be receiving a new treatment. It's a little intraocular implant that contains ciliary neurotrophic factor, which from now on I'll call CNTF. Okay. So, since all of you are clearly ophthalmology experts, this review is probably too superficial for you, but I'll go over it anyway. So, retinal tissue, as you know, is an extension of the brain, so it's specialized neural tissue that converts photons or light into electrical impulses, which are sent through the optic nerve to the brain. And within the retina, you actually have over 100 million photoreceptors, so little neurons all across the retina that do this job. So, to illustrate, we have light coming in. The vitreous or the jelly of the eye would be like here, and then this is sort of the back of the eye. And the light actually passes through all of these layers to then get to the rods and cones. And does anyone remember the difference between rods and cones? Anything? Come on, people. All right.
Rods are two.
What's that?
I think rods are more numerous and they primarily detect, non-colored, black and white.
That's good, exactly. So cones are responsible for the color vision. They work best in bright light. They're responsible for your fine vision and the fovea. And then rods are typically what you think of as like seeing gray images at night when the lighting is dimmer. And then once these cells receive the photons, basically there's a series of interneurons, and then the signals get sent out to the nerve fiber layer, which eventually connects to the optic nerve. So again, we talked about cones versus rods. Of cones, you probably might remember this from science and whatever grade, that you see light as blue, red, and green. So there are 3 different types of cones that are sensitive to different wavelengths. And then within the fovea, there are actually no rods, and that kind of connects back to what I was saying about cones being connected with fine vision at the fovea. So this is just another image of a cone cell, and you can see this is a nucleus here, and we'll just remember this area of the cone is called the ellipsoid zone. That's not something you need to know from medical school, but it'll come back later. So what's cool about cones, which I didn't realize, is they're actually highly organized. So the retinal tissue, when you look, this is under contrast microscopy, you see at the fovea, they're tightly packed. And the number that's been cited in numerous papers is 200,000 per square millimeter. So they're very densely packed in a regular mosaic. Then as you move out towards the periphery of the retina, you have rods that start to intervene. So those are those tiny little things here, and the cones are the large cells. And that's important for our project, and I'll explain why later. The fact that the cones are in a predictable layout. So switching gears a little bit, talking about retinitis pigmentosa, so also probably not something you've heard about in medical school. It's kind of a rare, what we would call orphan indication in FDA lingo, but basically it's an inherited disease, so it's genetic. There are over 50 known mutations, and what happens in retinitis pigmentosa is you have death of rod cells, and eventually that leads to death of cone cells, and this happens over a period of many decades, so you can see, if this is like what someone with a healthy eye would see, eventually someone with retinitis pigmentosa will see kind of tunnel vision because the only cells that are still functioning are some cones that are left in the fovea. And just another illustration, this is actually from a person that has RP, and they made a YouTube video. So this is just, again, showing kind of what someone with RP might see. There's also a lot of visual noise, like flashes, floaters that aren't really captured in this image. But essentially, it starts off with night blindness, and then you start to lose peripheral vision. You wind up with this, not so clear tunnel vision. Eventually, you lose all of your eyesight. So I'll just to show you clinically what you would see. Again, this may be review, maybe not. But we have the optic nerve here, the retinal vessels, the macula is this area, and then the fovea right there is what you fixate on when you read. And then in RP, you see some retinal changes. So you have, in general atrophy, you see these pigment deposits, the dark color here, and then changes in the optic disc as well as the vessels. So this is like a mid-stage disease here. So why we're studying this disease has to do with the fact that treatments are very limited. There was one RCT about 20 years ago that looked at high-dose vitamin A as a potential treatment because vitamin A does play a role in retinal tissue. But even though that did show some promise over the 4 to 6 year period that they studied it, the problem is, this disease, there are probably lots of different underlying mutations, some of which may be treatable by vitamin A, some not, and honestly there haven't been other great studies to confirm this. So some clinicians will recommend it, but it's not by any means definitive.
Sorry, so people do use vitamin A?
People will. So if you are going to take the high-dose vitamin A, there are potential issues with hepatotoxicity, so you need to be monitored. And also, for some RP mutations, like if you know your mutation, there are some that will not respond to vitamin A. So, the problem in trying to develop treatments for this disease is that, as I was saying, it takes decades for the person to have a clinically measurable change in their vision. And, we can look at their visual fields, we can look at ERGs, which are kind of analogous to an EKG in the eye, so it's measuring the electrical impulses in the retina. But those take years and years before you would see a potential difference between a treated eye and a non-treated eye. So we'll talk about what we're studying and then how we're going to measure it differently so we can do our trial in a shorter period of time. So this CNTF is the drug. It's a cytokine growth factor, and it's really cool actually. I'll pass around samples of the implants that we're using. Okay, so you can see it in these little capsules as a sample of the implant. So the implant is within the eye, it gets implanted by a retinal surgeon, and it has live retinal cells in it that will secrete the drug over a period of a couple of years. And the drug then kind of makes its way to the retina. With eyes, anything you put on topically does not have great penetration into the back of the eye, and that's why we're doing this as an implant. So CNTF has been studied in phase 2 trials in about 130 patients. And, like I was saying before, in the endpoints of this trial, there were no change in visual acuity, and that's not surprising just because this disease progresses so slowly. So there was one arm that was a treatment group, and the other arm received sham surgery, which I'll talk about. And because our design is going to use the same treatment and control arms. And these subjects actually did show decreased visual field sensitivity, meaning, in the treated eyes, they actually, it showed like their retinal cells weren't working as well in the eye that received the implant, but that was reversible after the implants were taken out. So overall, not hugely supportive of CNTF, but again, it's a 12-month trial, not long enough of a time point that we would expect to see measurable differences. So the way that the sham surgery is done, because a lot of you guys have asked, like, how's that possible? So normally when the implant is put in, there's an incision at the what's called the limbus, where the conjunctiva and the cornea come together, and then the implant is inserted through that. But in the sham surgery, a similar incision is made, but the eye is not penetrated. We don't enter the globe, and then pressure is just applied to the eye where the X is to simulate the movement of putting the implant in. So that's how the subjects are masked to whether or not they're receiving the treatment for a sham surgery. Does that make sense? Any questions?
Can you actually see the implant? Like if you were to look in the mirror?
Not with pupils that haven't been dilated. So that's part of the masking too. So our research question, given all this background, is in patients who are randomized to receive the CNTF implant in one eye and sham surgery in the other eye, will there be a difference in cone survival at 12 months? So we haven't talked about how one could possibly measure cone survival, but that's the new measure that we're going to be introducing to look at this drug and evaluate it in a short time frame. So again, we're doing a 12-month RCT that's double masked. We're going to use 8 patients with RP or Usher syndrome, which is a syndrome that, for our intents and purposes, the eyes have a manifestation of RP. And so that's actually, when you think about it, 16 eyes. Each person will have one eye receive the treatment and the other eye receive the sham surgery, so each person serves as their own control, which is a really nice way to eliminate confounding in our study. And again, you guys have seen the implant, but it doesn't interfere with vision, and you can't see it unless the pupil's dilated. For our subjects, we're going to give them the option to remove the device after the study's over. Like I said, traditionally with clinical endpoints in ophthalmology for this disease are not super helpful if you want a short trial. We're going to be using a new tool called adaptive optics, scanning laser ophthalmoscopy, and I'll explain what that is here. So this is actually a picture that we obtain of the retina with this new tool. So this is in real time. We're looking here, those bright dots are actually cones. So this tool has incredible resolution when you think about it. This is a picture of SLO without the adaptive optics. This is when you add adaptive optics, which basically is a technology borrowed from astronomy to reduce interference as the laser moves through the eye. So you get a crisper image, and then when you average a bunch of these images, you get a really nice overview of the cone mosaic that we saw earlier. And we're doing this in living human eyes, so someone just puts their head up to the camera and we can get this kind of image of the back of their eye.
Was this taken at like the fovea, or was this more peripheral?
Yeah, so what we're going to be showing are essentially foveal images because you get that clear cone mosaic, but that's a good question. So we're looking at the central retina. And I can show you, this is actually a real-time image of what we get. You can see, we're asking this person to hold their eye steady, and that's the image that we obtain when we have the patients place their heads up to the camera. And we'll ask them to look at different points in space to image different pieces of the retina. So we get a bunch of these videos, and we process them into still images like you saw in the prior slide. Then we put all of those together to create a montage of a piece of their retina. So this is actually one of our study subjects. You can see right here is the fovea. We have some nice blood vessels coming in, and then you can see the individual cone cells. So it's pretty cool because the resolution is just incredible. And to give you more perspective, this is the same montage superimposed on that patient's fundus photo. So it's a really small portion of the retina, and it's right near the fovea. And clearly, with just regular photography, you cannot get this kind of resolution. So when we get this montage here, we pick certain areas that we're interested in. We don't analyze the whole thing, and we call those regions of interest. We try to make them evenly spaced out throughout the montage. And then what we do is have those regions of interest analyzed to look at the cone spacing, with the idea that in this disease, as cones die over time, we would expect cone spacing to increase because there are fewer and fewer healthy living cones. And our graders, so we'll have 3 graders per region of interest, and they're masked and independent to help basically account for the fact that this is a manual process that involves subjectivity. So again, same montage. It's hard to see these little black boxes, but those are regions of interest. Again, phobia, region of interest. This is kind of blown up to a bigger picture, so we'd be counting cones in here. And manually, we place these red dots on what we know are cones. And the fact that the cones are arranged in a regular mosaic is important because it helps make this more reliable. You know, we know where we expect to see them. And then after that part is done, we do a calculation of cone spacing, which basically looks at, for any cone, what's the average distance between this cone and its nearest neighbor, this cone and its nearest neighbor, and it kind of sums all of that up. So we do have an extensive list of inclusion and exclusion criteria for this trial, which isn't surprising because it's a clinical trial evaluating an implant that involves surgery. And also a lot of these inclusion and exclusion criteria are set so that we're able to get quality images using the Oslo. So there are a number of things that will impact our image quality. For example, if someone has a cataract and not a clear lens, it makes it very difficult to get any kind of image resolved. Similarly, if they have, other ocular diseases, they may have retinal changes that we're not interested in for the purposes of this trial. So here's our timeline. So I'm lucky because I'm coming in at a point where we're just finishing collecting the 12-month images for these subjects. So they've already received the implants, they had their baseline imaging, their 6-month imaging, and now we're finishing with 12-month imaging. The primary outcome that we're interested in is the cone spacing, which is not typically, as I said, a measure that's used in ophthalmology clinical trials. Our statistical plan, so I'll just talk quickly about the effect size. So this is the size, the change in cone spacing that we expect to see over 1 year. And that's based on a paper actually done by a prior CTRFB fellow who looked at this. And arc minutes is like 1/60th of a degree of the retina. I don't know if you know the conversion of microns, but it's basically a small change in spacing. And we're going to control for the distance between those regions of interest that I said, the distance between them and the fovea, because we know that simply just being farther away from the fovea can change the cone spacing, and so we don't want that to introduce error into our outcome. We will still look at secondary outcomes, like this nice eye chart, otherwise known as visual acuity. And then I'll touch on one of those briefly, which is OCT. So that stands for optical coherence tomography, and this is a technology that is used in clinic to look at the health of the retina. So it uses infrared to image the retina in cross-section. So before we were looking at it on face, and now this is cross-section. So here's the fovea. And then what we're interested in is this layer that sits there, right here. And that corresponds to the ellipsoid zone, if you remember from way back before, that portion of the cone. So you can see it highlighted there. But we know that this reliably is a good marker of disease progression in RP. So that's why we're going to be focusing on that as a secondary outcome too. So I talked about some of our study limitations. The image quality can be a problem if you can't really resolve the cones well. We have seen for some patients at the 6-month imaging, they still have inflammation from the surgery. So people can also develop cataracts either post-op or just have, their regular cataract increase in density. You could see some edema in the retina. We do have a small sample size of 8 patients or 16 eyes. So far, this kind of study has only been done in 5 eyes, so it is a nice increase from prior. 12 months, even though we're using a super sensitive outcome, it's still a short period of time for this disease. And then we have to make sure that since each person's other eyes, their control, that both of their eyes have about the similar amount of disease at baseline. And we actually, I'm not sure if I put it up here, but that's part of our inclusion-exclusion criteria to make sure that that's the case. And then one thing we don't know is whether the simple act of the surgery or the sham would have any effect on the natural history of the disease. So that's an unanswered question. So next steps, we're just finishing up the 12-month imaging. We'll do that cone spacing, we're putting the little red dots everywhere, calculating cone spacing, and then do the statistical analysis. So, acknowledgements, mostly Jackie Duncan, who's been a fantastic mentor and has helped get me up to speed really quickly. And then the rest of our team here has been awesome as well, so. All right.
Questions for Stephanie?
You mentioned that cone spacing was like a relatively new sort of outcome measure.
Yeah.
Have people looked at using contrast spectroscopy or anything else, looking at people with RP and whether like cone spacing is even something that you would expect to see increase?
So the issue is if you're trying to do it in a living eye, this is the only way to do it, is with Oslo.
But have you seen, but like, they can see it in eyes of patients who,
Yeah.
I don't know, I guess who pass away from?
We've looked, so there are 2 things. One, is this even reliable in healthy eyes? And that's been investigated to be the case. So we know that it's been validated as just a general measure. And then looking in with microscopy in donor eyes, I don't know if anybody has done that.
That's a great question. There have been studies in non-human primates that have looked at non-retinal degenerate eyes, and there have been pretty good correlations between en-face microscopy studies and images with adaptive optics. But your point is really well taken because it's a relatively new technology, so there haven't been that many donor eyes available. We have one that is being studied right now. It's not the best quality histology study, unfortunately. Like the eye was not acquired quite as fast as I wish it had been, but the point is that it is sort of in the validation stage, and so we're hoping that studies like the one Stephanie's doing will help validate this as a useful clinical outcome measure that has high sensitivity and it's objective and non-invasive, and you can do it longitudinally. So it's got a lot of advantages, but your point is really well taken. It hasn't really, nobody has done great histology slides to confirm that exactly what we're seeing corresponds exactly to the slides, except it has been done in non-human primates and in rodent models.
So I was just wondering whether or not it's really like why you guys are doing the sham surgery, because it seems like there should you can't really fake the with the Oslo the results, so
Sure.
the placebo aspect isn't as clear to me, like what you're trying to achieve with the sham surgery. And I don't know the risk of I'm sure you guys thought about the risk of the sham surgery
Sure.
versus the benefits.
So I'll answer the risk question first and then why we're doing it. So the risk is really small. We're making an incision in the conjunctiva, which is just the membrane. It's kind of, the way I explain it to people who don't have medical knowledge is it's kind of like getting a cut on the inside of your mouth that heals itself. The risk of it, what we really worry about in ophthalmologic surgery are things like endophthalmitis or infection inside the eye, and that can't happen if we're only making the incision on the conjunctiva. So that's the main thing. Even the risk of the implant surgery is not that high. The reason why we're doing it, and I struggle with this too, is, when you think about the fact that we're manually identifying these cones, if someone were not masked to which eye received the implants and which didn't, then it can introduce a lot of bias in what you call a cone and what you don't. So does that make sense? So like, you know, you could be thinking like, oh, I see a cone here, I see a cone here, I see a cone here, kind of unconsciously because you know this is a treatment eye. So we wanted to mask the entire team to which eyes had received the surgery, and you know, we see these patients physically, so if the eyes have differential amounts of inflammation, then it's pretty clear which eye received the implant or not. I don't know if you would add anything
Well,
else.
the only other reason to mask the patients is that the primary standard outcome measures that are considered to be reliable outcome measures for every other clinical trial in ophthalmology are subjective, and they include visual acuity and visual field sensitivity. So we're also making those measures as part of the validation process of this trial.
Okay.
Not clear that 1 year will be long enough to identify a significant difference, but we certainly don't want patient bias to creep into those measures. So that's one reason to keep the patients masked.
Yeah, especially think about being asked to read the side chart.
Yeah.
Any other questions?
So I was gonna ask, are there other diseases that the osmo would be good for?
Yeah, so you're looking at it in macular telangiectasia right now.
Yeah.
And then,
Well, it's been
you know,
used for
there's
lots
a
of
whole,
things. Yeah, it's been used for pretty much anything that affects photoreceptors. It can also be really sensitive tool to look at retinal vascular perfusion, so things like diabetic retinopathy, branchial vein occlusion. It can be used to look at the lamina cribrosis that's being looked at in glaucoma models as well. It's good for anything that is reflective in the retina. So most of the retinal layers are clear, which is why we can see so well, despite the fact that the photoreceptors are at the back. But the inner segments with this particular wavelength of light do generate a signal. So anything that affects the photoreceptor inner segment mosaic is worth looking at. RPE cells can be seen with special wavelengths of light, and then retinal vascular changes. So macular telangiectasia, diabetic retinopathy, vein occlusion, things like that.
anything else?
You worried at all about the post-surgical inflammation? And like, if you had to redesign the study knowing now that even at 6 months you still see, would you maybe think about doing like maybe even more, like less sham and more actual device but minus drug in the other eye?
So you're asking a question about have we considered having the control arm just be an implant without any drug
Well,
in it?
if you now 2020,
Yeah.
hindsight 2020.
Yeah, so I mean the issue with that kind of boils down to what Nicole was saying about the risk of surgery, and you know, even though it would kind of be a better control in a lot of ways, it just, the fact that it introduces a greater risk of infection and bleeding and, you know, retinal detachment and cataracts, those are the things that we'd be concerned about. So especially, you know, these subjects already have limited vision, so we have to be really careful what we do to their eyes. But you're absolutely right that it would be a better scientific
It just seems like
control.
having
Yeah.
one eye inflamed for half of your child time
Yeah.
already introducing kind of a
Yeah, so I think the 6-month data it's gonna be interesting
Yeah.
to look at. I think the 12-month will be more, you know, accurate in that the inflammation will be less, but we don't entirely know. Okay.
You may have answered already. I was just curious how long it takes basically from like getting the images to having the measurements for one patient.
A while. I mean, the manual processing, like if you think about, this is essentially done mostly manually, so starting with these videos and then moving to the montage, I do this in Photoshop. So that takes like at least a week per eye, because I'm still slow. And then, you know, doing the cone spacing, I haven't even learned that technique yet. So we're working on getting it to be more automated, but it's too new at this point.
Great. Well, thanks so much.
Thanks, guys.